questions from the talks. So up next, we have our own Ehsan-Nejad from Dr. Reza Shadmir's lab. You're muted, 1 second. I will start the timer for you and I can unmute you.
So I start the timer for myself too. Okay. Thank you very much. I appreciate if people can turn on their cameras just for a second. So my name is Ehsan Sedaghat-Nejad. I'm a 6th-year PhD student from Dr. Reza Shadmir's lab. The work that I'm going to present today is a collaboration with 2 other PhD students in the lab, Paul, Hage, and JP. So most of the work will be a collaboration type of thing. So before I start going to the kind of science and what we did, I want to establish the concept of the internal models of the movement. So what I want you to do now is that imagine yourself that you're in a highway. It's nobody's around, so everything is safe. You are on the right lane and you're driving your car, or maybe driving your bicycle if you're not driving a car. I want you to imagine yourself and put your hands on the wheel. Okay, so I can see your hands. Put your hands on the wheel. Okay, and I want to ask you to close your eyes and change your lane in 5 seconds. Okay, so after I said go, please do that. Okay, so put your hands on the wheel, close your eyes, and change your lane. 1, 2, 3, go. Okay, great. So most of you are now off the road, kind of sorry for you. And the reason for that is, as far as I know, one person was doing this. How can you move the wheel with respect to your body? I mean, that's kind of amazing. But anyway, the reason is that you were thinking the car is moving in parallel. But what actually is happening is that the car will change its angle when you rotate the wheel, meaning that you have to rotate it back to correct the angle and make it back in the same lane. So it's actually an S-shaped movement. So this is an example that your explicit understanding of the dynamic of this car is actually incorrect, but nevertheless, you are able to drive very much correctly. So your brain has an internal understanding or internal models of this movement, and it's perfect doing it perfectly. Okay, so this was a very high level type of task, and let's do another very simple task that we are actually doing in the lab. So what I want you to do now is just track this dot on the screen. Okay, so let me see your eye movements. Okay, great. So what you were doing is that you were just looking here, looking back and forth between those 2 things. If I ask you what you're doing, you were saying that my eyes were here and my eye landed here and then I went back. But what happened in reality is that actually your eye will land a little bit short for kind of these high amplitude saccades. And then there's always a little bit of corrective saccade as what we call it. So even for this very simple ballistic eye movement, and what happened in reality is a little bit different about what you think it is happening. And that's what we are studying in our lab, about how does these internal models being implemented in the brain and what is the neural substrate for those sort of things. So from the patient studies and also animal lesion studies, it has been established that the cerebellum is the locus, is a potential part, is actually very important for the implementation of internal. So this is a patient study. This patient has a cerebral degeneration, meaning the Purkinje cells in the cerebellum has died. Can you see what is wrong with his eye movements? So whenever he makes the left far saccades, the eye starts fluctuating. But and also whenever he makes the right saccades, the eye fluctuates, but it's able to kind of converge. But a healthy control subject will make a saccade a little bit short, what we call hypometric saccade, and then correct it again, a short, and then correct it. Okay, so this is what will happen if these internal models, the neural substrate that encode these internal models will be damaged. Okay. So what we did in lab is that in 2018, I started working with marmoset monkeys and building this electrophysiological lab in our lab. So our lab was kind of just doing human psychophysics, but then we started working with marmoset monkeys. We first established the protocols. So this is the first time that these monkeys were being trained for doing this type of tasks. So we established protocols to train the animals to do saccadic eye movements. We also established protocols to do the head implant surgery. And very importantly, we established protocols to target deep brain areas so we can advance our electrodes deep in the brain and record from cerebral cortex. We also, because we are working with Purkinje cells, next slide, you will see an example of Purkinje cells. We also from ground up wrote a Python package software with a very cute GUI. And I'm proud of this GUI that we're using to sort the neural stuff. So let's see a sample kind of data. So this is a monkey. Instead of just making left and right eye movements, the monkey is making saccades in 8 directions, and then again another 8 directions. So it's a kind of 2-step saccade task. Here for you, I'm presenting the onset of the eye movement and when the eye movement is finishing. So these Purkinje cells that we are studying in our lab have 2 types of spikes. One of them is simple spike that's more similar to other spikes in the brain. This is a raster plot, meaning each dot is representing as happening of a 1 spike. So when it's sparse, it means that there is a decrease in the activity. When it's dense, that's an increase in the activity. Here is the estimated firing rate. So what you can see here is that these Purkinje cells have encoding for the eye movements, but it's not obvious what the encoding is. It's very complicated. Sometimes it's bursting, pausing, some other time it's pausing. And this has been the challenge in the field to understand what these Purkinje cells are doing, what the heck of this complicated system is doing. What we added to the kind of literature, and it's kind of what we're doing, trying to wrap it up and publish it, is that since we had the software and everything, now we can also sort the complex spikes of the Purkinje. So these complex spikes are complex, so they're really hard to sort, but with our things, we would be able to do that. And complex spikes are kind of sparse, but if we now look at the probability of a complex spike happening before the saccade, what it gets revealed is that these complex spikes has really beautiful tuning for the direction, okay? And this is indeed what we are going to use to make sense of this simple spike activity, okay? So first of all, let's see if we can replicate the literature and see what we can see the same thing that has been reported in literature. So if I put all of, so this is a population activity. So I put all of my 138 cells that we recorded from, we put in one basket and see the lump sum total activity of all these cells. What we'll see is that there is, as a population, they are encoding the saccadic hyphen. They will burst before the onset of the saccade, and they will pause during the saccade. But what is strange is that for different amplitudes or different peak velocities of the eye movements, these cells are not changing their activity. So it looks like they are modulating for the saccade, but they don't care if whatever type of the saccade it is. It's just firing the same way for all type of saccades. But if you now use this new framework, so if you look at these saccades not in terms of the absolute coordinate systems, but in terms of a relative coordinate system, and we define this relative coordinate system based on the tuning of the complex spike. So we are saying that this direction is a saccade onset. So now we put all of these directions for all cells together. Now, if you look at the activity of the simple spike, what we were able to see is that now we will see not only the encoding for the saccade, but also for different amplitude and different peak velocities of the saccade, these cells are encoding the ongoing saccade. So these cells, with this, we can solve this issue that how these Purkinje cells are contributing to the ongoing movement. We had to use the complex spike framework to be able to see their encoding of not just the saccade, but also the amplitude and also peak velocity of the saccade. Okay, just to wrap up, today I start with this very simple context, our concept of what does the interior model is. We did a very fun experiment with just simple saccadic eye movement and saw that even this simple eye movement can be different than what you think it should be. When the cerebellum get damaged, all types of control of movements also get damaged, and people may have very difficulty. In our lab, we established protocols and proved that the marmoset monkeys are very promising animal models to study cerebellum and also internal models of the movement. We developed a software package to sort the cerebral neurophysiology. And lastly, our kind of science-wise part of the talk is that using the complex spike framework, we would able to, I should play this for myself, we would able to make sense of the cerebral activity in the brain. Thank you, and I would like to take your questions.
Okay, great job. I see we have a question from Paul. Do you want to go ahead?
Sure. You mentioned that it was important to sort out the complex spikes in addition to the simple spikes, and I was curious, like, what advance exactly allowed that to be possible?
Yeah, so that's a great question. Let me go back a couple of slides so you can actually see. So uh, it's basically something named nonlinear dimensionality reduction, a technique called UMAP. So it's an advanced, it's a modification of TSNE, if you have heard of that. But using the UMAP, we were able to actually detect just a tiny differences in a waveform, and we were able to sort the complex bugs of the cerebellum. So these complex bugs are very hard to sort because their waveform can change even from one spike to the next a little bit, and that's enough for making the PCA and kind of linear algorithm kind of obsolete, but the nonlinear algorithm was able to sort them out.
Okay, and can we turn to some feedback from Joshua?
So that seems pretty cool. I'm curious, what fraction of the behavioral variance can you explain with your new model that includes complex cells, as compared to what fraction can you explain without that?
Yeah, so basically without this model, we cannot explain any variability in the data. So because it looks like that these complex bugs are just doing the same thing no matter what the type of the saccade is, what the amplitude is. So it depends on what you define as a variability. If you mean peak velocity, so our model is able to define the, it has a linear relationship between the peak velocity and the peak firing activity here. It also has a linear relationship between the amplitude of the saccade. So in that term, it's kind of very, very good. I cannot put a number on it because it's just a correlation to show that this model can has a very good correlation in predicting those things. Did I answer your question?
Yeah, thanks.